Welcome to BIO Asia-Taiwan. I'm the CEO of Top Science Biotechnologies Incorporation. Here I'd like to introduce to you the activities and the services provided by Top Science Biotech. We are located in Shiding, a scenic town on the east side of Nangang, less than 10 minutes away from here. We're a company with global vision. Our patents covers Taiwan, USA, and the major countries in Europe and China. We have been doing business with customers across Taiwan, Canada, Australia, and South Korea. As a startup company, we are honored to win the Golden Torch Award this year. Our businesses are based on a strong foundation of biotechnologies developed in-house. Currently, we are focusing on 4 areas. The first, reagents for cfDNA and cfRNA amplification and sequencing. The second is DNA sequencing and sequence data analysis. The third is laboratory technical support. And we also distribute consumables manufactured by our biotech partners. We have made a few reagent kits available in the market, including PCR kit, RNA PCR kit, and mRNA PCR kit. Recently, we have also developed a technology coined as GeneNet digital PCR, and protocols for one-drop of blood PCR, and for forensic DNA STR analysis. Here I'd like to give you a review of a publication we made in 2017 in scientific reports. In the paper, we compare the efficiency of PCR and that of the method used by Illumina. So as shown on the right-hand side, the PCR method is able to amplify the target DNA, here the cfDNA from a healthy individual. No matter how low the target DNA concentration is, the PCR is able to amplify all the target DNA molecules. On the contrast, Illumina's method failed to amplify the target DNA if the input DNA quantity is 0.05 nanogram or less. So under the low concentration, the method amplified the adapter dimers instead. So from this piece of information, we are able to expand the PCR method into the digital PCR. As mentioned above, we have developed protocols for forensic STR analysis and for cfDNA analysis. For forensic STR analysis, we are able to manipulate the genomic DNA from a single touch. For cfDNA analysis, we just need one single drop, about 10 microliter or even less, of body fluid sample without pre-isolation of the cfDNA. Coincidentally, you may have seen in the news that a research team in Cheng Kung University, they are able to use a single drop of blood for the validation of the efficacy of a vaccine. In parallel, we are able to use a single drop of blood for a complete cfDNA analysis. You may wonder what the GeneNet digital PCR is. Here I'd like to use a few slides to give you some idea about what it is and how it works. In short, the GeneNet digital PCR is a novel approach allowing us to catch almost all the cfDNA fragments within a defined genomic range. For more accurate estimation of the copy number variation, CNV, and for obtaining all mutation sites by sequencing. For a positive signal to occur, 3 events need to take place simultaneously. First is the target DNA must be there, and second, the gene-specific probe has to be there as well. And the PCR reaction has to take place. However, for the cell-free DNA fragment, since they are highly fragmented, many of them may not contain 2 primer binding sites on the same fragment, and thus they are called as negative, false negative. Moreover, there is no independent method to validate the CNV produced by digital PCR, and the maximum number of mutation sites can be detected by the digital PCR. It's 4 or less. To overcome the problem, we adopted the Bio's QX200 droplet digital PCR system to develop a GeneNet digital PCR method. First, all the components for the PCR reaction are mixed thoroughly and then vortexed with oil, and the preparation is subjected to digital PCR reaction. After digital PCR, the droplets are lined up and the fluorescent signals are detected to determine whether it's a positive or negative. Notice that many PCR methods can be tested for the digital PCR reaction. This slide shows in more detail about how the digital GeneNet digital PCR works compared to the conventional digital PCR. The conventional digital PCR adopts a Kerry-Mills PCR approach, which requires 2 primer binding sites. On the other hand, GeneNet digital PCR combines the conventional PCR with adapter-dependent PCR. As shown on the bottom of this slide. Okay, the purple track represents the target gene. And as shown on the bottom, Bio's method uses a B forward primer and a B reverse primer together with a B probe in between to detect the presence or the not presence of the target fragment. So they focus on a narrow spot within a few hundred base pairs. On the other hand, the GeneNet digital PCR, as shown on the top, uses A spot and a C spot to determine the boundaries of the net. So for the A spot, we have A forward primer followed by A probe. For the C spot, we have C reverse primer followed by C probe. And in the middle, we have a bidirectional primer, as shown here as B. B is an oligo, which is used to generate the adapter. So B is a bidirectional primer under this situation. So it can prime leftwards or prime rightwards for the PCR reaction. And so we can recruit all the fragments for the downstream analysis. The preliminary data has shown that the GeneNet digital PCR is able to increase the sensitivity by 2-fold. Here I use a flowchart to further illustrate how the GeneNet digital PCR works. First, we have the cell-free DNA, and the cell-free DNA is pre-amplified for the 2 purposes. One is to generate sufficient cell-free DNA for the downstream analysis, And the second is to build a construct to have adapters ligated to both ends of the target DNA. After preamplification, we use a GeneNet digital PCR for the analysis, and the preparation is then split into two aliquots, one for CMV analysis, another one for sequencing. Through sequencing, we'll be able to identify all mutation sites, and also by mapping of the sequence reads against the genome, we will be able to identify to get a number of mapped reads for the experimental gene and for the control gene. So by the simple comparison, we'll be able to obtain a CMV value based on the sequencing results. And this CMV value can be used to compare the CMVs generated from digital PCR. Here I'd like to announce that we will publish the complete Taiwanese monkey genome in the near future. And the one-drop blood protocol for cfDNA analysis is going to be available to the public in the near future as well. Here I'd like to thank my long-term collaborator, academician, Professor Alice Yu, and Professor David Smith at UC Davis, Professor Jiang Yu at National Chang'an University Medical Hospital, and Professor Curtis Pei at MPUSD. They are my collaborators on the monkey genome sequencing project. I'd like to also thank the people working in my lab, including Dr. Li, Lutimba, Ms. Zheng, and Mr. Xie. Thanks for your attention.